Hi everyone, and thank you for joining in for the Synthetic Biology Club hosted by the Abdel-Mawgoud Synthetic Biology Lab. This monthly online events, as usual, aims to promote discussions around synthetic biology and also to share its latest scientific advances. The format of the event is the following. So we'll start with a 40-minute, up to 40-minute presentation, followed by a 10-minute period of question and discussions. As a reminder, I would ask you to keep your microphone turned off through the presentation and also to keep your questions for the end. And before we start, I will let Professor Al-Masaleh to introduce our presenter.
Thank you so much, Benjamin. Thank you. So we have today the pleasure to present Dr. Vivek Murya, a professor at the University of South Korea, at Hallym University at South Korea. He will be talking about his article, Homology Modeling and Virtual Screening of P protein in a Quest for Novel Antimelanogenic Agents, published in 2014, Experimental Dermatology. Dr. Vivek Murya is an industrial biotechnologist. He worked at several universities and institutions of international reputation. Currently, he is a chief scientist at the Center for Energy and Environmental Sustainability in India, chief technologist at the Agriculture and Forest Conservation Research Center in Nepal, and team leader in Gangnam Sacred Heart Hospital, Hallym University in Seoul at South Korea. He also has worked as an assistant professor at Inha University in South Korea and at Sam Higginbotham University of Agriculture, Technology and Sciences in India, and also worked as a chief scientist at SS Measure Technology, a private company in India. And he also worked as a scientist at the Department of Science and Technology at the Government of India's Policies Research Center, and as a researcher in Gorakhpur University in India. Thank you so much, Dr. Murya, for having accepted an invitation. And it is yours now. Thank you. So I hope that you see the share button available in front of you, Dr. Murya. Your microphone is still muted.
So there is some glitch to share this. I mean, I don't know why my screen is not.
So if you'd like, you can just send me now the presentation and I can share from my side. And then you say to me next whenever you want to shift the slides.
I guess now it's okay.
Yes, it's fine. Wonderful.
So I just want to share one story about how, I mean, we got certain compounds which is related to anti-melanogenic effect. So our idea was not to develop the skin whitening agent, but obviously, I mean, industry is looking for the certain kind of the compound which is having more commercial value instead of the some minor I mean, our focus was to develop some compound which is helpful for the melasma disease. Melasma is very you can say how can I say, unpleasant skin condition where a dark patches occur without having any pathology. Instead of the, I mean, pathology, it has some psychological effect that people feel that, I mean, they are not looking good and having some odd and in situation when they go to certain kind of the social gathering. So we decided to find out certain kind of the target which not, you can say, interfere with the other biological process. While you can say most of the anti-melanogenic or melanogenic suppressing compound, it play intervening role between neurological I mean, you can say processes also. So that's why, I mean, we decided to find out certain kind of the protein which is exclusively find in your melanosomes. And it was the story of 2012 when we started to work over this project. And at that time, there was not much information related to this protein, and that was very new protein at that time. But nowadays, I mean, a lot of work has been done up to, you can say, this particular protein. So as I mean, I already discussed about, I mean, you already saw that this paper has been published in 2014, and obviously we publishes regularly, I mean, certain work over there. But I just want to share one interesting story related to this particular idea. I mean, you will get this particular picture where we know very well in a human, it's very unique, tendency because of the geological distribution as well as certain kind of the genetic, you can say, distribution. The skin color have a spectra of this. I mean, you can say color. It's mainly based on the combination of the U and pheomelanin. So melanin, I mean, when we talk about the eumelanin, it's really black or brown color. While we talk about the pheomelanin, it's usually, you can say, that is it's towards the golden color or little bit brownish color. So when you see the people, I mean, from the Irish origin, you can say origin, or if you see the people from the African origin and people from the Southeast Asia, as well as if you see the people from the Mendelian and Mongolian origin. The skin color can easily identified, like Mongolian and Mendelian people have yellow skin that sometime we, in our ancient stories, we say that it's a golden people. When we talk about the Caucasian who came from the, I mean, just from the your Middle East to India and as well as go to up to Bangladesh, Burma, I mean, our skin is called as brown skin or whitish skin. When we go to Europe, I mean, obviously the skin is very much that is having the very less amount of the melanin. So obviously these people called as, I mean, fair or white people. So this is all about the this particular story about this melanin. And our idea, I mean, our group is very much focused for the development of the anti-melanin as well as some other skin bioactive compound. So this was one story of the anti-melanin, because that project was very big at that time. It was related to a global, you can say, challenge program. So you can see in this picture that skin darkness is one issue that is highly in demand of, you can say, that cosmetics industry. While in case of the skin disease, when we have the patches, so this kind of the molecule can give a freedom from the patches, or you can say melasma. So that's a two major domain, I mean, this application had. So skin whitening as well as pharmaceuticals. So that was the basic strategy. I mean, you can find, I mean, if this you can say screen is visible, so you know that the melanin synthesis is not something, not, I mean, you can say bad for us. It's always good for us. I mean, as much as melanin synthesized on your skin, definitely you are protected from the harmful effect of the extrinsic factors such as UV, as well as, you can say, your some pollutant and etc. Because melanin is a protein which help our protect. It's just natural sunscreen, you can say sun guard type of the system providing via the melanosomes. So this is very important that melanin is not bad thing, but unfortunately our society, I mean not only you can say one society but entire society, they think that if you are fair then only you will be, you can say, look beautiful. So we just mess up that beauty is along with the fairness. I know so many people will not you can say agree with this point, and even myself, I mean I don't like this kind of the concept, but obviously we are working in industrial biotechnology domain, so we have to follow the industry until unless some certain social revolution is take place. So we have to do this kind of the research. So I mean our main agenda was, if you see the depigmenting agent in market, so you will find that hydroquinone and arbitune basically right now people are using for the research purpose because it's not very much effective. Its molecular size is very big, so that's why people are not using this one. And another one is your kojic acid and vitamin C. The kojic acid and vitamin C, it's very light sensitive molecule is there. So obviously, if it has been exposed to the light, so it won't be more effective. And hydroquinone is, you can say, most commonly used molecule for the skin whitening agent. Wherever if you see any kind of the cosmetics which claim that it will make you fair, so obviously it will have the hydroquinone. This hydroquinone has so many side effects, especially if you are applying for the long period, then you will lose the skin sensation because it interferes with the tyrosine activity. And we know very well that the tyrosinase is a key player for the nervous transmission, or you can say certain kind of the motor activity in a nervous signal. So that's why we decided to have a new molecule as well as new target. So this picture is very classical picture about the skin, you can say, in response, skin response against the ultraviolet and how they synthesize this. You can say melanin. If you see, if you are able to see this one, I mean there are certain kind of these stages, and obviously stage 4 is the highly matured melanin, which has to be go to keratinocyte, and obviously the keratinocyte will protect yourself from the ultraviolet. Now, in case of the molecular level, we know that it have the MC1R, that hormone receptor is there, and then it is started with the CRE and Krebs cycle, which ultimately regulated by MITF, which is exclusively related to tyrosinase production. So here it's this picture is very, you can say, clear. Then we try to find out how many proteins are exclusively related to melanosome. Melanosome is a kind of the specific somal body, I mean the body of the melanocyte, which exclusively find in only in melano, you can say, site. There is no other cell has been reported until that time which contains about the melanosome. So obviously we thought whatever protein found in as well as over the you can say membrane in melanosome, that will be exclusively specific for the melanocyte. So we try to find out, and obviously we find that there are so many domains. I mean, if you see that is clearly related, like lysosome, then another one is your secretory vesicle. So obviously these are the certain, you can say, common phenomena they had. And finally, we found that there is one, you can say, pH regulatory as well as melanocyte specific protein, which could be target for the synthesis. So among these key player, we identified that these are the key player. I mean, you can see that like the tyrosinase is the one of the fundamental player. Then tyrosinase related protein 1 and 2, then obviously other proteins. That is the very important for you can say synthesis of the melanin. So we go with the P protein, and we try to find out what is the role and the structure as well as, you can say, sequence of the P protein. So we try to understand this particular P protein, and at that time, it on the basic information whatever we had, it was transmembrane protein, and it had 833 amino acid. It was published in Genomics and Pigment Cell Research at that time. And one thing is very important that there is report, if there is a mutation in this protein, the people have, you can say blue eye. I mean, blue eye, usually we thought that the blue eye is the one of the, you can say, significance of the beauty. But the mutation in P protein also caused the blue eye, as well as certain, you can say, mutation in P protein. It reflect to albinism. And most important that these people who have the OCA2, I mean this P protein mutation, after certain age they got the pigmentation also. So it's not like that kind of the albinism, though throughout the life they don't have the pigment on their skin as well as eye. So we thought this could be a safer target. So that's why, I mean, we decided to just focus over there. So this protein was at that, I mean, it's a 12 membrane, 12 domain. I mean, it have the 12 barrel, which is a transmembrane domain protein, and highly conserved throughout the vertebrate, you can say, evolution. So we thought this is the very important, because this could be a target, and the C terminus, as well as you can say 2/3 of the human and mouse homologs, here 90% amino acid sequence identity. So we thought, I mean, this protein could be, I mean, this protein have some uniqueness because it's only found in melanosome, whether it's a mouse or any kind of the vertebrate animal. If it have the melanosome, then this protein is there. However, when we try to find out what, I mean, from where this protein came into melanosome, then we try to make certain homological study with the bacteria and some other organism. So based on that, we find that it is involved with the certain kind of the, you know, transportation. And I'm not going much detail because once we have certain summary that its function is not very much clear, but at that time, report suggests that the tyrosine transport is one major function. However, it also involved in regulation of the pH regulation of the melanosome, and pH is very key player for conversion of the, you can say, tyrosine to eumelanin, that means the cysteine to eumelanin. So that was the one major phenomena. And another one is that tyrosinase maturation is one more important phenomena. And eumelanin and pheomelanin content was another phenomena. Now we have to prove it. I mean, until unless we don't have, you can say, a concrete base to start this work, because there is no much information about the bioinformatics related to this particular protein. So we went certain, you know, you can say literature search and etc. Now, I mean, we come to two approach. One is genomic, another one is proteomic. So the proteomic information was not there because the protein structure was not there at that time. And you know very well, in 2012, the MD simulation was not very easy job at that time. Now it's very easy. I mean, every lab has at least, you know, the computer. So we have to make some interaction with some other university where the specialization computer was there. So we decided first we have to focus for the genomic approach and then proteomic approach. So for genomic approach, we try to find out what kind of the natural variant has been reported, where was the certain mutation was there, I mean, based on the genome analysis. So we find that there are so many type of the variations reported on NCBI. And after that, we go with the, you can say, analysis by protein sequence analysis. So we found that, I missed one point. Here we found that in case of the analysis that OCA2 is related with the certain kind of the albinism, or you can say lowered skin color. If the people have white skin, they have, you can say, non-functional P protein. So it means the P protein is directly related with the certain kind of the melanin synthesis. So we started with the, you can say, related to protein sequence similarity search. And we found that, yes, this protein have, you can say, any kind of the transporter, because it came, I mean, when we try to make the blast, we found that it have similarity with the transporters, but we don't know what kind of the transporter it is. So we try to find out what was the, I mean, the best possibility. So we, among all the search, we found that tyrosinase transporter activity is also there. Then we decided, make a, you can say, design a inhibitor which should have the tyrosine one domain and another one is arsenic inhibitor domain. So we just try to mix two molecules. One is tyrosine, assumption is that it will bind to the, if it is tyrosine transporter, it will bind to the, you can say, protein. And if it has certain degree of, you can say, like a transporter activity. So because arsenic inhibitor was there, arsenic transporter inhibitor was there. So obviously it will help to inhibit the P protein. Now, to validate our, you can say, analysis, so we go with the, you can say, homology search, and based on that, we try to find out where, I mean, you can say, similarity is there. And based on that similarity, we found that, especially, you can say, you can see that the highlighted domain, it had the mutation which is already reported, and it's related to the Black population, and that Black population, you can say, shown albinism. So obviously we just thought, because the tyrosine is very important molecule for synthesis of the melanin, and if it is non-functional, or it is not involved in the transportation, so obviously the tyrosinase will be not able to convert tyrosine into melanin further. So we go with that, and obviously, I mean, we try to do this kind of the study, and ultimately we found that, I mean, using the certain, you know, back references, and our idea was, make sure that yes, the P protein has certain degree of inhibition. So for that, we just took the siRNA, I mean, siRNA for the, against the P protein. So we transfected the siRNA using the liposome mediated. It was the, I mean, that was that lipofectamine, brand name is there. So we use that lipofectamine, just add the siRNA, which we synthesized in, I mean, through the company. And we try to find out the dose dependency, and let's see that whether it's working or not, because we use the cell that is under the cell culture, but we were not able to perform experiment at that time related to animal, because you know very well that animal testing is perfectly banned, or you can say completely banned. If you are trying to develop any cosmetics, we have to perform experiment on cell line only. That's the, you know, global, you can say, scenario. So we have to perform everything on cell line. So we started, you know, the melanin content in the melanocyte, and we find that yes, dose dependent, you can say, variation is there, and obviously the light content is high, and the cell is dividing every time. So the we cannot show the maximum, you know, that siRNA is present in every cell. So it's a total counting. So that's why we thought that this dose dependency is just like that. So we went for the, you know, different type of the concentration, and we tried to find out whether this siRNA is also, you know, making certain kind of the mortality. So we find that up to 40 nanomole there is no mortality, and our study was confined up to, you can say, 2.5 to 5 nanomole. So obviously this the safety concern was also, there was not much affected. Then we perform the, you can say, our basic experiment with the electron microscopy using the melanosomes. So you can see that this is the melanid, or that is the melanocyte. Here you can see that different stages of the melanosomes. I mean, that the dotted structure was there. And the scale bar was, we just increased the scale bar, and we find that the, you can see that here the mature, I mean, the perfectly dark balls, you can say, these are the melanosomes. And here the melanosomes were not very much mature. So we know that the inhibiting the P protein, the melanosome contain melanin, but not in that amount. It means the P protein is not only factor which is involved in synthesis of the, you can say, melanin. That means if we completely inhibit the P protein, still there is, you can say, required melanin will be synthesized. So that's why we thought it will be a very good kind of the target to sustain a sustainable inhibition of the melanin synthesis. Then also we try to quantify the you can say, U and pheomelanin ratio, and we find that yes, the U and pheomelanin ratio based on the HPLC. We try to find out what is the concentration. So we once we go with that, then we find that yes, the concentration of the U and pheomelanin is still, you know, in the same ratio, and that means the pheomelanin is not very much affected as compared to the control. So we went, you know, to this is the this particular will give you the basic idea that it lowered the eumelanin. However, the pheomelanin is not affected. That means your skin will be still, you can say, fight against the ultraviolet. However, the black spot can be easily down. So this was the, we also measure that the effect of the P protein siRNA on tyrosinase activity. So obviously the tyrosinase was not matured because we got some information that it also helped in tyrosinase maturation. So the tyrosinase, once we treated with the RNAi, so it was very interesting that the tyrosinase activity was inhibited. However, once we perform the Western blot assay, then we find that the tyrosinase detection was based on the dose dependency. So it was not there. When we can we see that, I mean, this is the tyrosinase specific monoclonal antibody. So obviously, I mean, you can see the concentration. But once we go with the other protein like TRP1, tyrosinase related protein 1 and 2, which is very important because it is involved in melanin synthesis. So that was not much affected. So that's TRP1 and TRP2 was not affected. It means tyrosinase protein is still there, but not matured protein. So this was the very safe target, I mean, in melanosome. And further we go with the, you can say, housekeeping gene, what kind of the effect is there. I mean, once we are inhibiting the protein, we found that while inhibiting this, especially based on the RT-PCR assay, we found that in case of the control as well as this treated one, the housekeeping gene was not very much affected. It means it is not going to alter most of the biological function. Then we again analyze other things. Obviously, I mean, we are going to reduce the melanosome activity. So certain kind of the like tyrosinase and TRP1, TRP2. So overall, I mean, tyrosinase here, when we go with the RT-PCR, it's almost similar. I mean, but in case of the Western blot, it is not visible because of the, it's a pro-protein. So then, as I already told you that how to design the inhibitor, this was the idea we developed in our mind, that let go with the, tyrosine as well as you can say inhibitor. So you can see that we try to use the tyrosine along with the inhibitor. We just replace this one with the tyrosine. So we got a new kind of the molecule, and this molecule, I mean, the structure of the tyrosine and uracilic acid, we just combine it. And after combining, we try to go with the, homology modeling. And after homology modeling, we perform the docking of the compound in the, you can say, barrel, which was made in a transmembrane domain. So we found that the affinity was very nice, and based on that, you can say binding score, we selected 25 different molecules. That molecules was downloaded from the database, that zinc database, and we try to find out how much was possible to you can say synthesize as well as you can say commercially available. So as we don't have the chemistry, you can say synthetic chemistry department in our you can say building, so we decided to purchase commercially, and we find that 20 molecule was able to you can say get commercial purchase over there. And then we screen these molecules, and we try to find out what kind of the inhibition they show. And these molecules were very effective because not more than 20 micromole we consider. And we found that up to 20 micromole after screening, we got 5 molecules, but one molecule have given certain strange behavior, because 4 have given all 4 have given a competition like P protein inhibitor, while one is performing certain different, though inhibition was there, but obviously this molecule is going to bind with some other transporter protein, because as I already told that we took the arsenic inhibitor molecule one domain, and another one is the tyrosine molecule. So obviously it go and bind with the certain other you can say pump, or you can say some other this transporter. That's why we I mean at that time we were so much pressure, so we just removed that molecule from our further study. And these are the molecule which has been already published and also patented over there. And then we find that, yes, here you can see that as compared to siRNA, when we see that in compared to siRNA, we found that this number 1, number 4, number 8, and number 13 was very, I mean, you can say nicely inhibited this P protein, and we got very significant result. And the result was proved by this, you can say, and then we further go with the molecule number 4, which was P protein level, and it was very interesting that it's a down regulation of the P protein was very high. However, once we go with the, you can say, Western analysis, the expression was perfect, and then finally we got certain, you know, conclusion which has been published, almost 4 papers in international repute, as well as 5 patents from this work. And right now, I mean, companies are working to formulate for developing cosmetics. However, we are also looking for some pharmaceutical application of this, but definitely requires, so, I mean, you can say some safety concern for developing the pharmaceutical, and some regulations are there. So this is all about the, you can say, work which we performed over there. And these are the patent which has been already, you can say, granted from the Korea itself. And I'm not sharing the publications because it's available on NCBI, so everyone can get these publications. So I think I'm on time and I finish this talk. So if anyone have
thank you, Professor Murya, for your amazing presentation and very interesting presentation. And I think now we can open the discussions and question session. So feel free to ask your questions and comments. You can use the raise your hand function, so we'll be able to give you the right to speak, or use the chat box if you're not able to ask verbally your question. Yes, Ahmad, you can ask your question.
Thank you so much, Dr. Murya, for your very nice presentation. It's really interesting, the fact that you investigated one protein and succeeded really to find some chemical inhibitors. But my question here is, what was the approach of your selection of P protein? Because I saw that you've listed about more than 10 proteins that are key players, that you call them key players in the melanin production. What was the specific basis of the selection of P protein? Is it about specificity? Is it the most unique protein in the melanin synthesis?
Yeah, I mean, you were talking about these, you can say, selections, right?
Exactly.
So we try to, find out the important, see, there are 192 genes which is involved in synthesis of melanin synthesis. That has been already reported. Now, our main job was to, mining of, gene from this 192. So first we have to, our first criteria, which is exclusively found in melanosome, number 1. So, and it's a direct involvement in melanin synthesis. So we find out that these, you can say, proteins, which is reported over here. That was our another study. I didn't share that study over here. But it was very simple thing. Number 1, that first of all, how many genes are involved? Then we have to go how many genes exclusively reported from the melanosome. Then we took these genes and go for the blast that, whether this gene is reported in other, you can say, organelles. For example, the tyrosinase. The tyrosinase gene was perfectly similar to the tyrosinase, which is involved in our neurological, processes. Because this melanocyte is also kind of the neural crust derived cell line when our development is there. So there are so many things common. I mean, obviously this is the integumentary system, so a lot of gene has been you can say mutated, attenuated during the course of development of the body. So we decided, let's find out which protein or which gene is unique in terms of the other organelle. So we found that out of all, because already skin whitening agent which we have, that is targeting the tyrosinase. So number one, if we are targeting tyrosinase, we will not get anything. So that was the very biased, exclusion from our study. After that, TRP1 and TRP2 we cannot do because it is directly involved in neural nervous system. So that was the you can say discarded. DCT and PMEL is also found in some other, you can say, organelles, especially you can say lysosomal mediated trafficking is there. Wherever lysosome mediated trafficking is there, I mean, like phagosome or any other kind of the biological activities there. So that was the discarded. Then we go with the, you can say, MATP. We found that the MATP, which is involved over here, it is very common in entire our body. So that's why it was not possible. MART also same problem is there. However, expression level is definitely higher in case of the this protein into melanosome as compared to other cell. But if we are getting anything new, then definitely it will be better. However, we got this P protein, and I am focused for P protein. I told my another student to focus over the SLC 45A2, but unfortunately we are not able to find out suitable inhibitor for this protein. So that study is another way. But selecting the P protein have two major idea, because literature suggests that it have certain function in you can say melanin synthesis, but this protein is very much unique. It is not found in any other, you can say, cell as well as you can say organelles. So that's why we decided, okay, let stick over there. That is the P protein. So unfortunately at that time, we were not, it was almost more than 10 years. So at that time, MD simulation was not very, I just saw your profile, your club profile. So right now it's very easy to you can say get the simulation and find out the binding energy, recalculate and minimize. But at that time we had certain softwares, and based on that software and also some free software, we just compare this, and based on the energy calculation, that binding energy, we just selected this you can say enzymes. So that's the idea. And you may also say that sometime your approach is, our bioinformatic partner told us this approach will not work. And we had a confidence, if this protein having certain biological function, then our approach will work. And once we got success in this particular approach, then they decided to find out the inhibitors, and based on that inhibitors, they go for the reverse, you can say, search related to stereochemistry. That is their part. So look for the stereochemistry of other biological molecules, and they try to find out the biological molecule which can inhibit the P protein. That's another story. They are doing that. So this was the major idea. Uniqueness, and another thing, the importance of this protein into melanin synthesis.
Very interesting, very interesting. So I think that based on your information, I think that if we are going to do that same study these years, we could just start first by comparing these proteins in terms of determining which of them is more impactful in melanin synthesis, to first define the target before we go to the search for chemical inhibitors by modeling and so on. So, for example, I like too much the experiment of siRNA, or we can now use CRISPR-Cas9 or dCas9 if it is just to, if many of these proteins are essential, so we can use dCas9 to compare these proteins and see which of them, while inhibiting them, will impact melanin the most, and then select the target that we will study to develop, for which we will study to develop an inhibitor, a chemical inhibitor. I think this is very interesting, and it's opening ideas now. So my second question is, your modeling was based on pure docking, and you have a list of chemicals that are supposed to have a potential to inhibit or bind to this P protein. Again, my question now for this point is, how did you check that? So how did you classify these? So did you have scores and you selected the highest score for inhibiting this protein, or you just relied on experimental? So you tested them all and then selected which experimentally is causing
yes, this is very valid question, and this question was very, I mean, we were very much scared, because once we make our molecule, we just make the molecule on ChemDra, and then similarity search of the molecule from the zinc database at that time. And exactly, I don't remember right now, but I guess it was more than 1,000 molecules was there. More than 1,000 molecule, and we try to make the library, and this library has been virtually docked over there. And after that, we had, I mean, you can say all the molecule and their binding energy was there. But we decided, let's go with the first 25, then second 25. This was the idea. But I was, you may say that I was lucky enough, because of the approach was very much right, that we took tyrosine and one transporter inhibitor and make a molecule. So usually what was the, I mean, at that time, what people are doing, they just took the zinc database and took all the library and just start, docking experiment at that time. So what was the failure rate? I mean, because, once they go with, you can say, calculation based binding, it was perfect. But after, biological screening, they failed. So every time, I mean, my bioinformatics partner from Yonsei University, they say that it won't work. I said, don't worry. I mean, I'm taking one molecule as a you can say, assume that yes, it is tyrosine transporter. So one is tyrosine, which help this particular inhibitor to get, you can say, interaction. That was one hypothesis. Another one with very strong arsenic inhibitor, you can say, domain. So if inhibitor domain is there, then definitely it will disrupt the, you can say, barrel structure. I mean, this 12, you can say, transmembrane domain, which is forming a pore, that will be distracted. So other, you can say, tyrosine will be not able to, you can say, enter into melanosome. This was the hypothesis. So based on that hypothesis, once we are designing our molecule, the success rate is usually higher. So whenever, any student asking about this kind of the work, so I always suggest them, you have to think like, you don't go, what people are doing. You have to find out what is uniqueness. You don't need to screen entire library. From that day to this day, the library is so huge. And if you start to doing all the library, definitely there are a lot of molecules which have the stereo, structure, which can easily bind with protein. But once you go with the, you can say, in vivo study, there are a lot of factors involved. Even our MD simulation is still so advanced as compared to docking, but it still includes, you can say, certain parameters. There are so many vital parameters which is not included and which directly affect, you can say, binding. So I suggest that once we are designing the inhibitor, we have to make one concept that this is the tyrosine. And obviously, if suppose, our plan B was also there, if it was not work, then we have to look for the another, you can say, inhibitor, transporter inhibitor, along with the tyrosine or tryptophan type of the amino acid. So let's find out any aromatic, you can say, amino acid combination, whether it's working or not. So luckily, we found that certain molecule which have the certain similarity with our designed molecule that was gave a certain binding score, and we took, first 25. Out of first 25, only 5 have given, you can say, positive result. And out of 5, one molecule we didn't consider as a P protein inhibitor. We thought it molecule have certain other kind of the inhibition also, because it reduces the P protein expression also. That's why we don't need that molecule, because we don't want to alter the P protein expression. We just want to inhibit the P protein function. So that's why we selected only 4 molecules, and that has been submitted for the, you can say, further study. So that was the idea over there. And I must say, right now, I mean, MD simulation is there. So our approach, I mean, if we use the MD simulation, so definitely the, you can say, getting the success chances is very high.
Yes.
Like right now I'm working on degenerative, you can say, approach, degenerative medicine, using the similar approach. So nowadays information is too much, and if we start playing with the information, I mean, obviously microarray data is there, RNA sequencing data is there. So if we start playing with the information, so we cannot get any conclusion.
Yes.
It's just like I mean, swimming in the mid of the that Pacific Ocean. So obviously you will not able to get any island. So it's better to, you know, focus for island and then move in that direction. So this is the one approach we opted, and we got this success. I mean, if anyone working in this idea, so I must say that your approach must be very peculiar, that you want a molecule. That's it. You don't need to explore everything.
Yes.
Because if we try to explore everything, I mean, we will not reach at any product. And being a biotechnology domain, I always say to my students that you have to think about the money, because if you will not think about the money, then you will not develop a product. So money in every matter, I mean, involvement, input cost, as well as whether you will be able to get royalty or not. If you are not able to get royalty, then okay, you have to publish your paper, then you have to go to biological division.
True.
So if you are in biology, you may explore the entire ocean. Definitely you will get a lot of pearls, but we are looking for getting, you know, some sandy area, sandy beach to reach at one point. So that's our approach. I mean, that's how we are working in Korea. This is how we are working. So that's why the patent is very important for in our research as compared to, you know, making big papers. So that's why the basic approach.
Thank you so much. Thank you. This is clear now. Thank you so much, Dr. Murya. That was my questions. Thank you.
Also, I think we've reached the 10 minutes of discussion. So this would conclude the activity. And I would like to thank everyone who joined and participated in the activity. And most importantly, thank you, Professor Murya, for your time and presentation.
Thank you. Thank you so much.
Thank you.
As a quick...
I must say good morning. Have a good day to all.
And as a quick reminder, if you want to present your work or article, you can always register as a presenter on our website. Otherwise, you can register as an attendee. And every last Monday of each month, we have a new presentation, and the next one will be on the May 30th. And we'll announce the next presentation the second week of that month. So goodbye, everyone, and have a nice day.
Okay, bye-bye.
Thank you. Bye.